What is glycogenesis? Glycogenesis simply refers to formation of glycogen. Okay, so how is glycogen actually formed within our body? So we just have two images. This is the first image which I'll be showing you. The second image which I'll be showing you, give me time to hide this. The second image which I'll be showing you in the next slide will be an image on how glycogen synthase is regulated and thus regulating this process of glycogenesis. So glycogen is a homopolymer found in the body composed of several units of glucose attached to each other by alpha 1,4 glycosidic linkages with branching points. So it is branched, but at the branching point, you have now the alpha 1,6 glycosidic bonds. So, how, how is it formed? It's actually a very easy process where you start off with glucose and phosphorylate glucose at carbon number 6 to get glucose 6 phosphate. So that reaction, which is not shown here, is carried out by the enzyme hexokinase. Then convert these glucose 6 phosphate to glucose 1 phosphate, and this reaction, which is reversible, is catalyzed by the enzyme phosphoglucomutase. Then take the glucose 1 phosphate um, and activate it. So how do you activate glucose 1 phosphate? You add a UDP, a uridine diphosphate molecule to it. So this activation of glucose is carried out by the enzyme or catalyzed by the enzyme glucose 1 phosphate urididyl transferase. This enzyme is also known as UDP glucose pyrophosphorylase. Okay, so these two are the same enzyme. Okay, now in that reaction you usually utilize UTP, uridine triphosphate, and generate a pyrophosphate. Then this pyrophosphate is cleaved to two inorganic phosphates by the enzyme pyrophosphatase. Now, the essence of this cleavage is to ensure that the reaction proceeds unidirectionally. Okay, you don't want this reaction to be bidirectional. You want to remove this pyrophosphate from the cellular pool and convert it to two inorganic phosphates. Okay, then you have now activated glucose. You have the active form of glucose here, which is the UDP glucose. So UDP glucose is the one now which will be added. Several UDP glucose are the ones which will merge with each other to form a glycogen molecule. But for that reaction to take place, where you are adding the UDP glucose, which is catalyzed by the enzyme glycogen synthase, you require a primer. A primer is required. Now, how is that primer made? That primer, for us to make that primer, we require a protein known as glycogenin. Now, glycogenin is a homodimeric protein, which has a molecular weight of about 37 kilodaltons. The important thing is it has a tyrosine residue at position 194. Tyrosine has a hydroxide terminal group attached to it. Now, it is to this hydroxide group that a UDP glucose is attached to by the enzyme activity glucosyl transferase. So, this protein, glycogenin, has the enzyme activity. So, glycogenin is an enzyme. Okay, so it has an enzyme activity there, glucosyl transferase activity. So, it carries out what is known as auto or auto glycosylation, self glycosylation. Okay, it adds uh, UDP glucose molecules to it. It attaches glucose molecules to it to get now these uh, primer here. Now this is the primer which has at least 8 uh, glucose molecules linked to it. Okay, so this is the uh, glycogenin, and you can see glycogenin is still attached to that oligosaccharide there. But then this oligosaccharide is attached to what is known as the reducing end of the glycogen molecule. So you only have one reducing end in the glycogen molecule, and to that reducing end, it's the protein glycogenin attached there. Now, the incoming UDP glucose molecules are attached to the non-reducing ends. So ends as in they can be many. you can have numerous non-reducing ends, but you only have a single reducing end for each glycogen molecule. Now, the incoming UDP glucose molecules are attached to the non-reducing end or the non-reducing terminal. Now, they are the incoming, the the carbon number 1 of the incoming glucose molecule is linked to the carbon number 4 of the resident uh, uh, glucose molecule. I have a feeling I've not said that right. Let me repeat that. Now, the incoming UDP glucose or the incoming glucose, it is the carbon number 1, the carbon number 1 linked to the carbon number 4 of the resident glucose molecule to form now the new alpha 1,4 glycosidic linkages. Okay, so glycogen synthase only makes the alpha 1,4 glycosidic bonds, but if there was no other molecule to act on this chain, this would not give us glycogen. Rather, it would give us a, a, a carbohydrate, rather a polysaccharide, which is found in plants known as amylose. So amylose does not have the branching points. It simply has those alpha 1,4 glycosidic linkages, okay? But glycogen, on the other hand, has um, branching points. So for us to make these branches, you require the branching enzyme. So the branching enzyme has the 4,6 transferase activity. In fact, that's the short form of the amylo 1, amylo alpha 1,4 2 alpha 1,6 glucosyl transferase. Okay, glucosyl transferase. So that's actually the activity of the branching enzyme. And see what it does. It takes an oligosaccharide chain from the non-reducing end. So it takes an oligosaccharide chain like that one. Ideally, it should be about 7 or 8. Okay, 7. It takes up that oligosaccharide chain from the non-reducing ends. So it cleaves this alpha 1,4 bond. Okay, and then it takes this uh, uh, oligosaccharide chain and attaches it to another glucose molecule, either of the same chain or another chain. Okay, so once, so in this case, you can see that chain was attached to the glucose labeled G. So labeled G there to form now a new bond, and this bond is now the alpha 1,6 glycosidic bond. Once you do that, you get now two non-reducing end, one there and another one there. What is the importance of this branching? One, you increase the solubility of glycogen. Two, you increase the number of the non-reducing ends. So when the non-reducing ends are many, you the process of glycogenesis and uh, uh, glycogenolysis can occur at a very fast rate. Okay, so if you have two non-reducing end, you can find at one end there is chain elongation, so there is glycogenesis. At the other non-reducing end, you have chain breakdown, you have glycogenolysis. So the same molecule of glycogen can be acted upon by two different enzymes. Okay, so further elongation takes place at the non-reducing end. This will be carried out by the glycogen synthase, which makes the alpha 1,4 glycosidic linkages. But branching will be carried out by the branching enzyme, which has the amino alpha 1,4, alpha 1,6 glucoside transferase activity. And by that activity, you make the alpha 1,6 glycosidic linkages. And by doing so, you get that branched molecule, which is a homopolymer of glucose known as glycogen. Okay? So this process of glycogenesis is a cytosolic process. All these enzymes we have just stated are exclusively found in the cytoplasm or the cytosol. Now, how is that process regulated? So you simply need to regulate the activity of the enzyme glycogen synthase. So glycogen synthase can occur in the A form, which is the active form, or the B form, which is the inactive form. So contrary to most enzymes of the body, most enzymes of the body are usually active in a phosphorylated state, but glycogen synthase is active in a dephosphorylated state. And inactive in the vice versa state. So the enzyme which phosphorylates the enzyme glycogenesis is the enzyme protein kinase A. So protein kinase A is activated by a molecule known as cyclic AMP. So in presence of cyclic AMP, the inactive protein kinase A is activated, and once activated, it phosphorylates glycogenesis to an inactive form. Now, cyclic AMP is generated from ATP by the enzyme adenyl cyclase. So adenyl cyclase is linked to a G protein. Okay, it is linked to the G coupled receptors. Okay, the G coupled receptors, the G protein coupled receptors. Okay, so those receptors, it is to those receptors that glucagon and adrenaline or epinephrine binds to. So these two inhibit the process of glycogenesis. So if you don't want glycogenesis to occur as one would require during starvation, these two molecules prevent that, and you can see the sequence of events. But in glucose plenty, when you want glucogenesis or glycogenesis, you want glycogenesis to occur actually, the enzyme insulin usually activate a protein known as protein phosphatase 1. And this protein phosphatase 1 dephosphorylates glycogen synthase. So by dephosphorylating it, you take it from the inactive state to the active state. Also, insulin activates an enzyme known as phosphodiesterase. And this phosphodiesterase uh, breaks down the cyclic AMP into 5' uh, AMP, and by doing so, the protein kinase A remains in an inactive state. Okay, so insulin upregulates glycogenesis, but epinephrine and glucagon downregulate glyco, rather yes, glycogenesis. Okay, so that is that. I hope this has been helpful, and follow for more biochemistry content. And thank you for your time. Asanteni sana.